Hello, my name is Mary Beth Babbis. I'm a clinical pharmacist, and I'm honored to be presenting an overview of neurobiology as it relates to psychopharmacology. You can see from our objectives that we are really looking at basic science, especially that which underpins pharmacogenetics, an increasingly important topic of psychopharmacology. When we think about basic science and neuropharmacology, we often just think of the big three that are involved in mood and cognition. The two catecholamines derived from tyrosine, namely dopamine and norepinephrine, and the one indolamine derived from tryptophan, serotonin, also known as 5-hydroxytryptamine or 5-HT. Before we get deep into the subject, quick review of action potentials. At rest, a neuron has a negative intracellular charge compared to extracellular charge. Negative resting membrane potential of about minus 60 millivolts. When ion channels start opening, a signal starts that causes the influx of sodium or calcium. The inside of the cell becomes more positive. If it reaches a threshold potential of about minus 30 millivolts, an action potential will ensue. This action potential can travel down the length of the neuron. If it makes it to the terminus, the change in voltage will cause the opening of voltage-gated calcium channels. The influx of calcium will cause fusion of the synaptic storage vesicle membrane with the nerve terminus to cause exocytosis of the stored neurotransmitter. The neurotransmitter then will diffuse and interact with a receptor for which it has affinity and activity to produce the action. If the action is opening of sodium and or calcium channels, then the result is depolarization, more positive charge of the receiving neuron. This is known as an excitatory postsynaptic potential. On the other hand, if the receptor action leads to opening of chloride channels so that the negatively charged chloride anion rushes into the cell, or opening of the outward flowing potassium channels so that the positively charged potassium runs out of the cell, that receiving neuron becomes hyperpolarized, more negative, less likely to reach that threshold potential. This is known as an inhibitory postsynaptic potential. So here you go. Which do you think is most likely to open a chloride channel, caffeine or a benzodiazepine like diazepam Valium? If you chose diazepam Valium, you're correct. They cause opening of a GABA-gated chloride channel, actually increasing the frequency with which that channel opens. Chloride rushes in, causing suppression of neuronal firing, pretty much in a broad action in the brain. Look at what we use benzodiazepines for. Preoperative sedation, including production of an anterograde amnestic effect, and anxiolytic, sedative, hypnotic, decreased seizures. At high doses, we might also see muscle relaxing effect. In other words, an overall suppression of CNS firing. It's a little bit more complicated when you're looking, at a single neuronal pathway. When we look at a single neuronal pathway, it's not a single neuron. It's a pathway. It's a complex of neurons, wherein if you excite an inhibitory pathway, what you get at the end is going to be more inhibition. If you inhibit an inhibitory pathway, what you get at the target effector organ or neuron is going to be more excitation. So sometimes you have to logic your way through the neuronal pathway to figure out what the end result is going to be. We need to also discuss the blood-brain barrier to help set up a basis for some of the most important pharmacogenetic points. So first, our CNS blood vessels are sealed by tight junctions between the endothelial cell with limited availability for paracellular transfer of materials. We have astroglial foot processes whose lipid bilayer membranes create a lipid barrier that decreases penetration of non-lipophilic substances into the brain. So only small, highly fat-soluble lipophilic substances can access the brain via passive diffusion. These astroglial processes or astroglial cells are actually metabolically active. They have extrusion pumps, efflux pumps like P-glycoprotein or multi-drug resistance protein, MDRP, that use ATP to pump xenobiotics and drugs away from the brain back into the bloodstream. They also have cytochrome oxidase P450 isoenzymes that we normally associate with hepatic phase 1 biotransformation, but they're present in other places. So single nucleotide polymorphisms, or SNPs, in the genes that code for these pumps and these enzymes can cause differences in access of substances to the brain. If the substance that doesn't normally get to the brain can get to the brain, then we see a change in pharmacodynamics. Another important point is the fact that only lipophilic substances can passively diffuse into the brain, Lipophilic substances have a greater tendency to undergo phase 1 biotransformation by CYP enzymes in the liver, another point of pharmacogenetic variability that can lead to alterations in pharmacodynamic response. Indeed, there are not really that many important pharmacogenes identified with regard to psychopharmacology to date. Here are 5: CYP2C9, CYP2C19, CYP2D6, and with regard to risk for cutaneous reactions, SNPs in our human leukocyte antigen coding genes, both for HLA-A and HLA-B, confer alterations in pharmacodynamic response, in this case, increased risk for cutaneous reactions. So pharmacogenetics can be an invaluable tool. And as a team, it's going to take all of us to educate ourselves, but also to educate and empower our patients to make the right decision when it comes to applications of genetic testing and medication selection. This data, what we know about pharmacogenetics, as we can see, can help reduce adverse drug reactions, as in the case of the carbamazepine and the HLA-B, but also can help guide rational medication selection. But we have to think about benefit to risk ratio, and we have to be able to communicate that benefit and that risk to our patients so that we can get informed consent before we recommend genetic testing. So one of the risks, one of the limitations that I see is not all findings from pharmacogenetic studies are really actually clinically relevant. So we'll look at some sources of guidelines that help parse through what is clinically relevant, what isn't. Another great limitation that I see is just really interpretation, because there are lots of different interpretations of these results. That's why use of guidelines is so important. Privacy and confidentiality, huge. While privacy and confidentiality are protected by HIPAA and GINA, there are huge gaps in the Genetic Information Non-Discrimination Act, GINA, that don't apply to the insurance industry. So genetic testing may cause higher premiums or exclude the ability of a patient to purchase life, long-term care, and/or disability insurance. There may also be discrimination by other institutions through employment. So it's another one of the risks that we have to consider. Equity and access, right? Is this a tool that's only available for those who can afford it financially? Does the data reflect a diverse population, or is it much more homogeneous and lacking, our ability to interpret it in the context of like these diverse populations? That's something I worry about. The basis of the research and the subject selection when the research was done can severely limit our application. There's also some misinterpretation and issues with clinical utility. I've seen a lot and heard from a lot of the med students that I've spoken to out in the field where they're testing for SNPs in the serotonin reuptake transporters. There is evidence to support that certain phenotypes have a decreased response to the selective serotonin reuptake inhibitors, compared to people that don't have those SNPs. But right now, the clinical evidence does not support application of that data. So, using an evidence-based set of guidelines can be very helpful. So this graphic is depicting those 5, common pharmacogenes. How you would interpret it is, I'm just going to pick the easiest one, which is at the lower right. We see the orange diamond. I'm sorry if you're colorblind, but that's what I'm saying. At the lower right of the diagram, we see that only carbamazepine is connected to the SNP in human leukocyte antigen A to have, clinically based evidence that impacts use of carbamazepine. This is a cut and paste from the Clinical Pharmacogenetics Implementation Consortium website, CPIC, but there's also the Pharmacogenomics Knowledge Base from GKB that is tied to CPIC that offers us information to help us interpret this in a clinical context. So we see that the second column is the protein coded for by the gene impacted by the SNP. The third column is a drug associated with that SNP in that particular gene and protein. The next column is a link to the clinical guidelines. This is probably the most useful thing. The next to the last column tells you the level of applicability of the guideline. actionable, meaning if you have the test and you have the outcome, then you need to take action. But notice that for HLA-B with carbamazepine, we actually see testing required. This is that Southeast Asian population with an HLA-B, I think it's a star 1801 phenotype, who has a very severe risk of Stevens-Johnson syndrome and or toxic epidermal necrolysis when carbamazepine is used, such that you would not choose that drug at all. The last column links to the PubMed ID for the study used to develop the evidence to make the guidelines. So it's a very useful website. I put the QR codes and the website names there for you. I would really recommend checking those sites out. Moving on to basic neuroanatomy and some of the key players, a quick review. The cerebral cortex from the Latin meaning bark, kind of connects many areas of the brain. It's a thin layer of highly myelinated white brain matter, that relays messages all over the place. And then our prefrontal cortex, the very foremost part of our cortex, is a section of our brain that controls behavior to allow us to live in a group society. And indeed, the size of a prefrontal cortex of any species is directly proportional to the size of the social group in which that species lives. I think that's kind of a cool idea. So anyway, the big neurotransmitters in the cortical areas of the brain are glutamate and GABA, with a lot of relay neurons, interneurons that are acetylcholinergic in nature. Moving on, and not so important as a direct target in neuropsychiatric pharmacology, but the basal ganglia often takes a hit when we use dopamine blockers in particular. The basal ganglia consists of three deep-seated nuclei, the caudate and putamen, which are together known as the striatum, and the globus pallidus. So regions of the basal ganglia help initiate and control voluntary movement, especially the involuntary portion of voluntary movement. The substantia nigra, black substance, stains dark on histology, is a dopaminergic pathway that arises in the midbrain and terminates in the striatum. This is also known as a nigrostriatal pathway. Degeneration of this pathway, of course, is associated with a movement disorder known as Parkinson's disease. The hypothalamus is involved in integrating information from hormones and neurotransmitters to help maintain homeostasis. The lateral hypothalamus secretes the wakefulness neuropeptide orexin, help switch us toward wakefulness during the wake-sleep cycle. The ventromedial hypothalamus controls sexual motivation and even probably sexual orientation. The thalamus itself, so hypothalamus, below thalamus, hypothalamus actually gates information, and during a wake-sleep cycle, it works in conjunction with the reticular system to help regulate sleep. So the reticular system, we have an ascending and descending part of this system. The ascending portion originates in the ventral periaqueductal gray matter, very heavily implicated in signaling by monoamines, dopamine, norepinephrine, histamine, serotonin, and also including the wakefulness peptide orexin that we previously mentioned. The descending pathway originates in the ventrolateral preoptic nucleus, VLPO. Think about the pineal gland and the influence of darkness on the secretion of melatonin. So melatonin, GABA, and galanin inhibit the wake cycle. And those are part of the descending reticular activating system. The limbic system is one of my favorite parts of the brain. The limbic system is not really a formal system, but is an older grouping of organelles consisting of the amygdala, hippocampus, and the cingulate gyrus. It's responsible for emotion, social behavior, pain perception, learning, and memory. I like to call it my monkey brain because it's a much more primitive part of my brain. And as I previously mentioned, it's controlled, well, not directly mentioned, but there is a direct control between our prefrontal cortex, PFC, and our limbic system. So our higher brain controls our monkey brain. Within the limbic system, the amygdala itself plays a huge role in the expression of anger, rage, fear-motivated behaviors, social cognition, and decision-making. The nucleus accumbens is a part, the group of structures, or part of a group of structures that are dopaminergic and that actually will terminate in that PFC, the prefrontal cortex. It's associated with reinforcing properties of reward, so food, sex, addiction. The hippocampus plays a huge role in memory formation and probably undergoes alteration as a result of stress. It's been found that constant exposure to cortisol causes atrophy of the hippocampus, which inhibits the growth of new neurons and impairs our ability to form memory. The limbic system, as I mentioned, normally controlled by our prefrontal cortex, wherein increase of dopamine in the prefrontal cortex inhibits dopamine release in the limbic system. Let's look at our neurotransmitter players now that we talked about some of our big pathways. We don't usually talk about pathways when it comes to our amino acid neurotransmitter because these buggers are everywhere. So when we have monocarboxylic acid neurotransmitters like GABA, like taurine, like glycine, like beta-alanine, generally you're going to evoke an inhibitory postsynaptic potential. Or dicarboxylic acid like aspartate and glutamate, much more likely to evoke an excitatory postsynaptic potential. Glutamate in particular is really important for long-term potentiation and long-term depression that allows for synaptic pruning for memory consolidation and learning. I'm going to diverge here just for a second to go into the monoamine theory of depression that posits that major depressive disorder is caused by a loss of dopamine, norepinephrine, and or serotonin at the synapses. Well, let's look at this. We put a patient on a normal, typical antidepressant drug, and they get side effects right away. 4, 6, 8 weeks later, they start maybe to get a therapeutic response. That tells me that there's more to the story than just the monoamine theory. Another piece of this puzzle is the use of NMDA receptor antagonists like ketamine, S-ketamine, that cause pretty rapid improvements in depressive symptoms. We do know that ketamine, S-ketamine, that NMDA receptor antagonists cause an increase in the production of brain-derived neurotrophic factor, BDNF. Okay, so there are polymorphisms in the gene that codes for BDNF that seem to confer a loss of therapeutic action of ketamine and S-ketamine, that rapid response, that rapid antidepressant effect. So I think the story is much more complex. Regarding our NMDA receptor antagonists, particularly ketamine and S-ketamine, there is a CYP2B6*6 allele that is associated with much more severe dissociative effects from these drugs. There's also a single nucleotide polymorphism in the norepinephrine transporter that is associated with much more significant cardiovascular effects, including hypertension from these NMDA receptor antagonists. Remember that when we antagonize the NMDA receptor, we end up increasing sympathetic outflow. So that explains the relationship to the norepinephrine transporter. Anyway, let's get back to the nitty-gritty of our glutamate receptors. The two that I'm going to focus on, but we have ionotropic receptors, those glutamate receptors that gate ion channels, and we also have metabotropic receptors that activate other pathways, including G-coupled protein receptors, G-protein coupled receptors. So let's look at AMPA and NMDA. The AMPA receptors fire at low levels of glutamate, and they are sodium and calcium channels that causes an increase in positivity of the receiving neuron. The NMDA receptor is normally blocked by a magnesium ion, but when that potential becomes positive enough in the receiving neuron, that magnesium pops off and allows for the influx of calcium. When the NMDA receptor is firing, it increases the expression of AMPA receptors. So there's this sort of positive feedback loop going on. The other thing that we need to know is that when we have lots of calcium in a cell, we have lots of activity, it generates lots of reactive oxygen species. Glutamate excitotoxicity is implicated in many neuro disorders, including reperfusion injury, post-stroke, Alzheimer's disease, et cetera, et cetera. So holistically, let's look at magnesium. One of the ways that you can tell a person is hypomagnesemic in the United States is that they are breathing. I'm sorry, that was kind of a joke, but really our diet is not, the magnesium intake in the standard American diet, the SAD diet, is woefully inadequate in magnesium, something that needs to be addressed. Anyway, I'll get back, I guess, to the topic at hand. GABA opposes the effects of glutamate. It opens those chloride channels, allowing the influx of chloride if it's GABA A, or it opens the potassium channels, allowing for efflux of potassium if it's GABA B activated by a drug like baclofen, generic name for Lyoricel. Interesting points. The relationship between glutamate and GABA. Glutamate decarboxylase is found in the astroglial cells. It depends upon the presence of pyridoxal 5' phosphate derivative, active derivative of B6. Think about isoniazid. We prescribe B6 to prevent neurotoxicity from isoniazid because in part of glutamate excitotoxicity. Moving on to our biogenic amines, we're really not going to talk about acetylcholine. I just want to bring up another nutritive point about neuronal firing. Notice that tyrosine, an amino acid, is converted to dopa, is converted to dopamine, is converted to norepinephrine, is converted to epinephrine. These pathways rely upon tetrahydrobiopterin, BH4, which in turn relies upon folate, B6, B12, and S-adenosylmethionine. We also see that vitamin C is critical for the conversion of dopamine to norepinephrine. My point is, you can't have healthy neural function if you don't have a healthy diet. Nutrition plays a key role in neural function. Dopamine pathways are really important. We talked about the basal ganglia, the substantia nigra. We talked about the control of the PFC, the limbic system. We need to look at the hypothalamus, wherein dopamine release, I guess we already mentioned that, inhibits prolactin secretion. We used to use dopaminergics to inhibit lactation postpartum in people who did not want to breastfeed. The flip side of that coin is in people who were assigned male at birth, you put them on a potent D2 blocker like haloperidol, gynecomastia is a real risk. Technically not in the CNS, but in the circumventricular area postrema, our dopamine receptors are kind of sampling the blood, looking for xenobiotics, and evoke an emetic response. In the periphery, our dopamine receptors are GS protein coupled. The GS in the smooth muscle causes smooth muscle relaxation. So in the GI tract, a dopamine agonist can cause relaxation of the lower esophageal sphincter, causing reflux, decreased peristalsis, leading to constipation, dilation of the renal and mesenteric arterial beds. Dopamine agonists can lead to postural hypotension and a reflex tachycardia. Again, dopamine release by the prefrontal cortex inhibits dopamine release in the limbic system to decrease the positive symptoms of schizophrenia. A point that we need to make, though, is that serotonin 5-HT2C receptors inhibit dopamine release in the prefrontal cortex. Okay, so serotonin inhibits inhibition of dopamine in the limbic system. Most of our atypical antipsychotics, well, actually, by definition, an atypical antipsychotic blocks those 5-HT2C receptors, inhibiting the inhibition of dopamine in the prefrontal cortex, which then inhibits dopamine in the limbic system. That's why we went through all those pathways. Really quick point about ADD, ADHD. It's an imbalance between norepinephrine and dopamine firing. So while these two molecules are very closely related structurally, the alpha-2 receptor is thought to increase the strength of the signal in ADD, ADHD, while activation of the dopamine receptor is thought to help dissipate the noise. Pharmacogenetically, atomoxetine, the norepinephrine transport inhibitor, severely increased risk of adverse effects in 4-2D6 metabolizers. Serotonin, melatonin, so they kind of flip-flop. In the daytime, serotonin's higher. When the pineal gland is activated, conversion of serotonin to melatonin is increased. But my point here is niacin deficiency. The disease of pellagra, including dementia. In severe niacin deficiency, some of the tryptophan that would normally form serotonin is converted to niacin. So again, you can't have a healthy brain unless you have a healthy diet. I'm a chart maker, so I made this chart, a summary of selected serotonin drugs. A couple of important points. Number one are 5-HT2A agonists, LSD, psilocybin, cause disruptions in thalamic gating. So sensory input becomes a bit garbled. But what we have found, or what they have found in some preliminary research, and there's continual ongoing research, is that activation of the 5-HT2A receptor may have some therapeutic benefit in conditions like PTSD. Another point, again, to reemphasize that our 5-HT2C agonists cause satiety, and there was once a drug known as lorcaserin for weight loss, are atypical antipsychotics associated with a risk of weight gain. And that brings me to our final point about us as a team, holistic medicine, and histamine from the tuberomammillary nucleus. Histamine increases the release of serotonin in our wakefulness pathways and also at that 5-HT2C receptor. When we put patients on drugs that are antihistaminic for a long period of time, we can get increase in appetite and weight gain. So it takes all of us thinking rationally about medication choices to help maintain the best health in our patients. So from a psychiatric practitioner standpoint, your involvement with your patient's primary care provider at the level of histamine and serotonin receptors can really have a big impact on their overall metabolic status. Anyway, enough said from a cross-field pharmacist. I'm at your service. If I can ever be of assistance to you, please email and I would be happy to help.